Dear colleagues, my name is Anastasia Ilyina. I am a research assistant of the Laboratory of Molecular Mechanism of Aging at the St. Petersburg Institute of Bioregulation and Gerontology. Today I'd like to focus on the mechanisms of regulating the functional state of the female reproductive system by short peptides. It should be noted that, against a backdrop of growing scientific and technological progress, there is an increase in psycho-emotional state in the background of chronic or acute stress, which in turn is associated with the development of neuroendocrine syndrome. The frequency of menstrual cycle expansion in adult women increased by 2 times, in adolescents more than 3 times. Changes in neurotransmitter systems of the brain were identified in the most common hypothalamic pituitary dysfunctions, accompanied by impaired ovarian function. Thus, disorders of the reproductive processes are the current issue, forcing specialists to look for new approaches to development of therapy. The basis for regulating the functional state of the female reproductive system by the peptide bioregulators is an integrated approach to influence 3 components, which are the biorhythm of sex and stress hormones, the functional state of ovaries, and the blood supply to the organs of the female reproductive system. We will discuss the properties of the biologically active supplements endolutein, Ventfort, and Gerontology, whose active components are IgG peptide, KIG peptide, and ovarian extract, respectively. The active components of the endolutein and Ventfort were developed in the study of epiphysis and vessel extracts from young calves, respectively. Firstly, let's consider the regulation of the biorhythm of sex and stress hormones. It is known that melatonin is involved in the formation of biorhythms, regulates the functional state of endocrine glands, body temperature, carbohydrate and lipid exchange, arterial pressure. There was a decrease in the peak concentration of melatonin in blood plasma and night excretion of 6-sulfathoxymelatonin, indicating a deterioration of the melatonin epiphysis in physiological aging. By this reason, there is an issue of correction the functional deficiency of the epiphysis with age. In this regard, the influence of endolutein on melatonin-producing epiphysis function was studied. The study was conducted on almost healthy elderly people who were divided into experimental groups according to the randomized principle. 15 people received endolutein intramuscular, 10 people received physiological solution. The control group consisted of 10 young men. Before and after administration of drugs, the concentration of melatonin in blood plasma was determined at 9, 15, 21, and 3 o'clock by radioimmune method. It should be noted that the distribution of people according to the initial functional state of the pineal gland showed that most subjects in this group had reduced melatonin production function of epiphysis and disrupted the daily concentration of melatonin in blood plasma. Therefore, the effect of the drugs was evaluated separately in subgroups of people with preserved and reduced melatonin production function of the epiphysis. Endolutein administration didn't affect the melatonin concentration in plasma of older people with preserved function of the epiphysis. The course administration of endoleukin contributed to an increase in melatonin concentration in blood plasma of people with a reduced melatonin production function of epiphyses at 21 and 3 o'clock. It should be pointed out that endoleukin didn't affect melatonin concentration in the plasma during daylight hours. Thus, the dependence of endoleukin's effect in the initial level of melatonin production function of the epiphyses indicates a modulating effect of this biologically active supplement. Cortisol is another important hormone in regulating the daily activities of the nervous, endocrine, cardiovascular, and immune systems. Therefore, the influence of endoleukin on cortisol production in macaques of different age was studied. The study was carried out on 6 young and 6 old females of rhesus macaques. Among the animals of each age group, a control group with infusion of saline and an experimental group with endoleukin injection were selected. The drugs were intramuscular administered for 10 days daily. Then, blood was taken from the elbow or femoral vein, which resulted in the release of plasma. Cortisol content in plasma was determined by immunoenzyme analysis. The result of the study showed that the administration of endoleukin to old animals restored circadian rhythms of cortisol content in peripheral blood. It should be noted that there is a non-pronounced decrease in cortisol levels in the union with aging, which indicates a reduction in the amplitude of circadian rhythm of this hormone. Since the epiphyses and the adrenal glands are closely related, it can be assumed that restorative effect of endoleukin on circadian rhythms of cortisol in all animals is mediated by the recovery of melatonin secretion. This is confirmed first by the existence of the negative correlation between daily cortisol and melatonin levels in peripheral blood of young macaques. Second, the circadian rhythms of melatonin and cortisol normalize when endoleukin is administered. Next, let's discuss the regulation of ovarian function with peptide bioregulators. The study was performed by female rats of the Vista line, whose cycle was determined based on cytology of vaginal smears. From the age of 3 months, female rats were transferred to conditions of food deprivation with free access to water according to the scheme. Day normal diet, day is hunger. This scheme was carried out for 30 days. Rats were divided into 7 groups of 10 animals per each. Intact rats didn't receive any effect. Rats of first and second control received the saline solution by intramuscular or intragastrical administration respectively. Third, fourth, sixth, and seventh groups of rats received genoleukin in therapeutic or 5 multiple therapeutic dose in different ways of administration. No differences in the length of the estrous cycle were found for female rats from experimental groups and group of intact before stress modeling was started. A reliable significant on average more than 2 times elongation of the estrous cycle was observed in females 30 days after the end of stress exposure compared to the group of intact and with a baseline in each group. The length of the estrous cycle in animals of the second and fifth groups didn't change compared to the previous period of observation after the end of 10-day course of the Gerontology administration or physiological solution. The duration of the estrous cycle in female rats of third, fourth, sixth, and seventh groups who received Gerontology at therapeutic or 5-fold dose for each route of administration was reliably reduced by an average of 2 times compared to before use in the same group and approached the baseline level for each group. The length of the estrous cycle in the female rats of third, fourth, sixth, and seventh groups wasn't different from the baseline in each group after the end of a 20-day course of the Gerontology administration. The duration of the estrous cycle in the female rats of the second and fifth groups was reduced compared to previous observation period, but didn't reach the baseline in each group and different from it, as well as from the indicator in the group of intact at the same time of observation. Thus, the results of this study suggest that a Gerontology as a polypeptide bioregulator based on extract from the calf ovaries tissues restores estrous function in female rats. There is a reason to suggest that Gerontology is a promising means for the prevention and treatment of pathologies of various origins caused by the impairment of functional activities of ovaries. Next, let's discuss the mechanisms of maintaining a normal blood supply in organs of the female reproductive system. The study was carried out using primary cell culture of Wistar rats vessels at the third and 14th passages, which were respectively taken as young and old cultures in accordance with the recommendation of the International Association for Cell Culture Research. In each of the passages there was a control and group with addition of biologically active compound went forth. Further immunohistochemical study was performed by primary antibodies to the signal molecules involved in the functioning and aging of the endothelium. In particular, the Ki67 protein is a well-known and widely used as a proliferative marker. The aging process is characterized by reaching the healthic limit and decreasing or completely stopping the cell's ability to divide. Therefore, the Ki67 protein is an important marker for evaluation the reduction of proliferative activity of cells and the degree of involution process in the studied organ. The results show that the area of Ki67 protein expression in the control young cultures exceeded this indicator in old cultures by almost 2 times. The Wentworth addition has contributed to a reliable increase in the Ki67 protein expression in both young and old cultures of vessel cells of rats. Next study was devoted to evaluation the influence of Wentworth on the expression of p53 protein in cultures of red vessel cells. The p53 protein is a transcription factor involved in suppression of malignant tumors by activating apoptosis in the tissues. p53 protein is activated by DNA damage as well as by stimuli that can lead to such damage or a signal of cell aging and disruption of its functional activity. The results showed that the Wentworth application to young cultures contributed to a decrease in the expression of the p53 protein compared with corresponding control. There aren't any changes in old cultures supposed to Wentworth treatment. Then, the effect of the Wentworth on the expression of signal molecules in culture of human endothelial cells was evaluated. Primary endothelial cultures from the vein of the human umbilical cord were received with the support of the Institute of the Obstetrics, Gynecology, and Reproductive Medicine named OTA. Cells were cultivated similarly to 3 and 15 passages. In each of these passages, there was a control and group with addition of Wentworth. On the next step, immunocytochemical study was performed by means primary antibodies to the protein Ki67 and P53. It was shown that area of Ki67 expression in control young cultures is more than 2 times higher than in old cultures of endothelial cells. When adding, the area of expression of Ki67 has been reliably increased in both young and old cultures with Wentworth compared to the corresponding control. The result shows that Wentworth stimulates a decrease in proliferation of the endothelium of the vessels with age. The proliferative action of Wentworth can be directed to repair vessels when they are damaged with age, as well as in pathological conditions. It was shown that the expression area of p53 protein in control old cultures exceeded this indicator in young cultures by more than 3 times. The expression area of p53 protein didn't change in young cell cultures of the human endothelium and decrease in old cultures with Wentworth treatment compared with the corresponding control. Thus, in the culture of human endothelium cells, Wentworth exhibit bioregulatory properties depending on their initial state. Thus, with the aging of the endothelium, there is a sharp increase in apoptosis. This indicates that aging in the body slows down the processes of cellular renewal. In this case, it is important to preserve the viability even of those cells whose functions are impaired. In this sense, the detected anti-apoptotic effect of Wentworth is a geroprotective one and allows to maintain the integrity of endothelial line of the vessels. Thus, a complex effect on functioning of the female reproductive system is possible by means of several peptide drugs as Gerontology, Endolytin, and Wentworth. A drug based on the extract from the ovaries, Gerontology, normalizes the functional state of ovaries. The drug based on the extract of the pineal gland, Endolytin, normalizes the biorhythm of the secretion of sex and stress hormones, which in turn directly influence on the functional state of the female reproductive system. A drug based on the extract from blood vessels sent forth regulates the functional state of the vascular endothelium, which affects the blood supply to the organs of the female reproductive system, including improving microcirculation of the ovaries. That's all I have to discuss in the present lecture. So, thank you for your attention.